Hello students, welcome to SK Biology. Today I am going to discuss the Lac-Operon, which is a classic gene expression and regulation model in prokaryotes. Gene expression is the process by which genetic information encoded in DNA is converted into functional product, generally a protein. In prokaryotes, gene expression is simple, rapid, and highly regulated, allowing bacteria to quickly adapt to changing environment conditions such as nutrient availability. Now, the question is how genes are regulated at transcription level. Answer to this question in the case of prokaryote is they uses operon model. Operon is functional unit of DNA. In bacteria, it consists of structural genes, promoter and operator, all expressed together as a single unit. So, single promoter is required to regulate expression of all structural genes. I repeat, single promoter is required to regulate expression of all structural genes. Examples of such operon model are lactose operon, tryptophan operon. We are going to discuss in detail lactose operon. Let us start with historical background. The concept of Lac-Operon first time explained by Jacob and Monod in 1961 while studying lactose metabolism in E. coli. Key contributions of Jacob and Monod are as follows. They proposed operon model of gene regulation and also demonstrated that genes are not always expressed. They also explained how gene expression is controlled by regulatory proteins. They are awarded Nobel Prize for their work. Their work laid foundation of molecular genetics and gene regulation. These are some important terms and their definitions which we are going to use while understanding lactose operon. Lac-Operon is a group of structural genes and some regulatory genes that regulated and expressed to ensure that enzymes required for lactose utilization are synthesized only when lactose is present. So, Lac-Operon is called as inducible operon and found in E. coli that controls the metabolism of lactose. Now, let us understand structure and components of Lac-Operon and respective function of each component. The first component is Lac-I gene. It is a regulatory gene which produces repressor protein. Second component is promoter. It is the site of RNA polymerase enzyme binding. Third component is operator. This is the binding site of repressor protein. And then next component is three different gene, LAC-Z, LAC-Y and LAC-A. We call them as a structural genes. These genes are actually transcribed into different mRNAs and then translated to synthesize specific protein. LAC-Z synthesizes beta-galactosidase protein. LAC-Y synthesizes permease protein and LAC-A synthesizes trans-acetylase protein. These proteins has specific functions as beta-galactosidase breaks down lactose into glucose and galactose. Permease is useful for transportation of lactose sugar and trans-acetylase is required for detoxification. Now let us understand very important point, that is how these components regulate lactose metabolism. There will be two cases. First one is in absence of lactose. All the regulatory as well as structural genes are present in DNA of E. coli. But in absence of lactose, when LAC-I produces repressor protein, this active repressor protein binds to operator region of operon and it restricts binding of RNA polymerase. Without binding of RNA polymerase enzyme, there will be no transcription of any structural gene. That is LAC-Z, LAC-Y and LAC-A. Without mRNA, no translation will occur. So ultimately, no synthesis of any protein, beta-galactosidase, permease as well as trans-acetylase. So conclusion is, In absence of lactose, Lac-Operon is switched off. Now, the second case is presence of lactose. All regulatory genes and structural genes are present in DNA. In presence of lactose, when Lac-I produces repressor protein, lactose sugar binds to repressor protein, which made some structural changes in repressor protein in such a way that repressor cannot bind to the operator. So, operator is available for RNA polymerase binding. As soon as RNA polymerase binds to operator, it transcribes all the structural genes producing three different mRNAs. These three different mRNAs get translated into three respective proteins, that is beta galactosidase, permease and transacetylase. So it concludes that in the presence of lactose, Lac-Operon is switched on. So, in prokaryotes, gene expression and regulation changes on the basis of nutrient availability like sugar. If glucose is available, cell will go for glycolysis, which is a constitutive pathway, the pathway which is always active in the cell. But when lactose is available, which is a disaccharide made up of glucose and galactose, bacteria will switch Lac-Operon system. So, the lactose operon is active only when lactose is present. Overall, in E. coli, lactose will be entered inside cell by permease enzyme. Then, beta galactosidase enzyme breaks lactose into glucose and galactose. Further, toxicity is removed by transacetylase enzyme. So, this is all about Lac-Operon when all genes are active. In next part, we will discuss what happens when any gene of Lac-Operon is get mutated.